again. Hello again. I'm back now, and I want to speak here now about in silico cloning, basically. So I come back again to my PowerPoint that I share with you guys. And yeah, before I show this slide, and in the following couple of slides, there are exercises about cloning. And as already discussed before, so I basically only use Golden Gate cloning and only use this hierarchical DNA assembly, which decomposes in the first place sequences into functional units, and then these functional units can be reassembled into genes. So let's say I want to change a promoter, then I just use a different promoter in an assembly, and I will get a different construct in the very end, driving a different expression level. So I said here, first exercise, cloning of PCR product encompassing a gene from Arabidopsis, its respective cDNA, into a plasmid called PICH 86988. So let's first of all try to get the sequence, the plasmid. So where do we get it from? We can get this from AdGene, because this is a plasmid that was deposited with AdGene. So I hope that everybody knows AdGene. It's a plasmid repository that, well, for the academic field. So we can deposit plasmids, and they send them out for the researchers to the researchers. It may appear expensive sometimes, but I think actually with these 70 euros or whatever it is for a single plasmid, it's quite fair. And even more fair are the prices for collections. So you can get 96 well plates filled with plasmids, and these will be like 300 or 400 euros, something like this. So you can get quite something for your money. Also, sometimes gene synthesis also in a similar level. So, okay, so we have this, I just took the name from my PowerPoint, and I say I want to look for this in plasmids, and I will find exactly one here. So if I click on it, I come to the plasmid entry page. I find here who deposited this plasmid, so Silvestre Marie-Launay in this case, where it was published, and we have here a link for sequences. So I can go on that. And what I get here is a full sequence from depositor. So this is what Silvestre Marie-Launay uploaded. And then I get a partial sequence from AdGene. And this means that they used a bit of Sanger sequencing to verify that this plasmid is really what it is supposed to be. So now all plasmids submitted or deposited with AdGene after date X, so everything of the past couple of years, actually doesn't have a partial sequence anymore. They are all resequenced using nanopore technology. So we have now a full sequence from AdGene with newer plasmids. So I can now get this one here. So I can just take a quick look, analyze sequence. This is what AdGene does for us. It shows some features like, for example, the origin of replication, kanamycin resistance gene, and so on and so on. But since we have actually our own software to display this, we don't want to work with their display. So I can download here for SnapGene or for GenBank. So I select the GenBank format. So depending on the depositing person, you can also sometimes have as additional data here and yet another sequence file, and sometimes the degree of annotation, the degree of information in these files can be quite different, because from this link here, you may get only limited annotations, while the second link may provide more. But this depends really on the case-to-case basis. So it's now in my download folder. And tick. Here, this one here, AdGene plasmid 48076. So I can again drag and drop this into my folder here. And it will now be called exported only. So I don't like that, because it will be chaos in a couple of minutes. So I can rename it. I can make a right click and say rename. I can also press F2, or I can just, as in any Windows File Explorer, press once. And then I will get into the editing where I actually have to press twice. Well, you know what I mean. Now I can call it PICH 86099. Be actually able to identify this in my database. I can say this this here, this was Pseudomonas aeruginosa A with some upstream downstream sequence, doesn't matter. And this was the whole genome of Pseudomonas aeruginosa. So I would not delete the original file identifier, even though it is still contained here normally in this type of information. It should be visible here. Well, it's not. It's somewhere. I just don't know where. So let's take a look at this plasmid. So plasmid is circular normally. That's at least what we're used to see. Let's put this in the circular view. And again, I do have my restriction sites displayed. So now my selection starts to make sense because, as I said, I always use Golden Gate cloning, normally using the enzymes BSA1 and BPI1, and these are now displayed. And this plasmid is also made for Golden Gate cloning. So basically, we have two BSA1 sites here that do flank a LAC-Z alpha gene under LAC promoter control, and this is used for blue and white selection. So basically, if we replace this BSA1 fragment with something that we want to clone into this plasmid, then this part will actually be eliminated and replaced by the gene, and the bacteria will grow out white on plates containing X-gal, while the original plasmid will grow out giving blue colonies. But what does our plasmid actually do? So this is a plasmid for agrobacterium, so for plant expression. And the part of the sequence that is transferred into the plant cell by agrobacteria is flanked by left border and right border tDNA repeats. And these are annotated here as a miscellaneous feature, MISC feature. So it means basically all of this will go into the plant cell if we use agrobacterium for plant transformation here. So let's analyze this content a little bit. So we would have a NOS promoter, a kanamycin resistance gene, and a NOS terminator. So this would be a cassette to select transgenic plants by kanamycin resistance. And we have here a second cassette. So we have a 35S promoter from cauliflower mosaic virus, which will drive strong constitutive expression in the plant. Then we have the cloning site, so this will disappear. And we have an OCS terminator, so a strong terminator as well, coming from octopine synthase from agrobacterium. So this plasmid is nicely annotated. We have a lot of information. We also see here that we have another kanamycin resistance gene. So this is now for selection of transformed bacteria. So once we have cloned into, we have made a ligation or something like this with this plasmid, we will select on kanamycin. And we also see that we have ORI-V and Rep-A of the PVS1 origin of replication in the backbone, and also this star A region. So this will actually be required for replication in agrobacteria. So in this circular view, it doesn't matter how far I zoom, I can never actually see the nucleotides. So if I want to see the nucleotides, I have to go back to the linear view, and maybe I highlight this little section before. So I just go here. So the beauty of Golden Gate cloning, as I said, is basically that the restriction sites are asymmetric and can be placed so that they are in one fragment or in the other fragment. So here, as I said, we want to replace this fragment that is flanked, that is containing the marker gene, so the LAC-Z gene, and by a gene of interest. So in this case, you see the BSA1 sites are inside, and they will disappear with this fragment. So if we have our insert properly designed, the final product will not contain a BSA1 site anymore. But, the The other thing about golden gate cloning and these type 2S restriction enzymes is that we can freely select this overhang. So if we want to clone something into this plasmid, we actually have to create such overhangs on our insert. So let's just go back to GAC-A. Let's say I want to express the bacterial response regulator GAC-A in plants. Maybe I want to do that. So I would now have to create an AATG overhang to be able to clone this in there. So normally in plants, well, in eukaryotes, we only have ATG as a start codon, but in bacteria, you have several ones. So now we would have to decide whether we want to add an additional methionine upstream of the initiating valine that is coming from the bacterial genome, or whether we change the valine into methionine for the plant expression. This is what I actually would do. So I would replace this codon by an ATG. So now we have a methionine, and this will be cleaved off anyways in the plant. I said we need an AATG, so we would need an A here, and we don't need any of this. So I just select it and press delete. Up, gone. So if I add the adapter, I can now just mark the A and add my restriction site. So it would be for BSA1, it would be GGTCTC. And then we have an N, and then it will cut a ATG as a 4-base pair sticky end overhang. Yeah, so an N for the software doesn't make any difference, but it's not good for primer ordering. It will be more expensive. So let's just put an A. Can be any type of nucleotide, but I put an A in this position. Then as well, the restriction enzymes generally like to have something to stick to at the end. So we put an additional 2 T nucleotides. Again, this is random on this side. This would be now something that would be a proper adapter to actually be able to clone this into this plasmid. So these adapters in the lab, I would add them by PCR. So they would be part of a PCR primer. So making PCR primers, you can make it complicated with many rules. You can make it easy by just looking at the sequence. And in this case, I would surely take the second option, because anyways, it has to be at the beginning of the primer. The oligonucleotide has to bind at the beginning of the gene if I want to make this fusion when put it into this gene, into this plasmid like this. So the only real variable is the length of the primer. So I would make it something between 19 and 25, let's say, and depending on GC content, I will decide between these different lengths. Important is, of course, that the adapter doesn't contribute to hybridization. So the part that is really complementary to the PCR template has to be like these 18 to 25 or 19 to 25 nucleotides. Let's make it 19. So this is the part that has to be contained in my primer for hybridization. This is the part that has to be contained in my primer for adding the adapter. So all of this will make my PCR primer. So I can again select this sequence stretch, make a right click, and select now here add annotation. I say this is now an annotation of the type primer. And I call this, it's my very first primer. I just started working in the lab. I call this 001. Actually, maybe like this, because I anticipate that I will order more than 1,000 at some point of time. So if I would make a PCR with this oligonucleotide on the Pseudomonas aeruginosa genome, then I would create an adapter suitable for cloning into this left-hand side. So I have to take care of the right-hand side. So here now we see a GCTT overhang. Okay. So where would we have to select this? So since this is just about expression of a gene, so now with bacterial gene in a plant, and I do not have a translational fusion or anything, I could theoretically put this anywhere. So, well, anywhere downstream of the GAC-A gene, but it also doesn't make sense to include additional genes. So I would place this overhang just here, and also my second cloning adapter. So to do this properly, I can select all of this and make a right click again and say edit selection. So I delete this because I don't want it to clean up my sequence. So my sequence actually stops with a TAG, stop codon. So I already have a G of a GCTT overhang. I don't need to add the G again. I can make CTT. Of course, if I add it once again, it doesn't change anything in life. Now again, I need to add also my restriction sites. So it was a GGTCTC. So now I have to put it in the other orientation. So it will be a GAGACC. And here at this very position, I actually need to add my little n. So I put once again an A in the other orientation. So I put a T here, and I put two A's here. Because I now did all of this on the top strand. Okay, so you see now I would have indeed a GCTT overhang that is cut by BSA1. Here's still the annotation of UVRC, so the downstream gene. Actually, it doesn't exist, so I can delete this. I can make a right click and say delete annotation, and it's gone. Now, once again, we have to make a primer. So once again, I use my simple rule of thumb 19 to 25. So this would be 19. There's quite a number of GCs, so there is no point in making it even longer. So I can select all of this, make a right click, and so this will be now my second primer. If I do it like this, it will be first in the wrong orientation, okay? So this is not the sequence, the orientation my primer should have. So I can make a right click again, edit annotation, and put it in the minus orientation. Up. Now it would be the right sequence. So there is a big difference whether the annotation is in one orientation or in the others. So if I select the double-stranded sequence view, you see that if I click on this one here, I get the upper strand selected. If I click on this one here, I get the bottom strand selected. So just by double-clicking and pressing now Control-C or saying, or mousing over the annotation and saying, actually, this doesn't work. Like this, copy. I get my sequence, and I mean to order nucleotides. To make a nucleotide list, I would always use Excel. So let's say name, sequence, and rank, just for fun. So I paste my sequence in here. This would be my forward primer. This, oh. Okay, wrong command. It's not length, it's only length. It's 29 nucleotides long, and I said this is JS0001. While if I double click on this one, I get this sequence highlighted, yeah? So if it's single-stranded, you don't see it, but it's still the same sequence. So it should start now with a TTGGTC. Actually, did I copy? I don't know. The thing is that if I select it like this, it will be the 5' 3' prime sequence as visible on the screen. So GGTCG. Completely different story. Yeah, so this actually you have to pay attention to this, of course. 31 nucleotides. So now I have a, so this would be like a PCR product, yeah, if I want to really clone this. There may be a command to directly make PCR products, but I'm not aware of it. So when I do primer design, I more or less do it like this. And now if I want to make a cloning of these two together, so this is pretty much the same as what we had in this little exercise that I put into the PowerPoint. Then what we do is to select these two files. So I can close both of them, it doesn't really matter. If I change this now, I will actually change these, save these changes to the original file that still had the upstream downstream sequence. Normally, I will not want to do that, so I say no. This is not like I lost my stuff now because I saved this as a new file, while this still is the original file which it derived from, yeah? So here in the history, we should see now that this comes from the genome here in the history. So I take the two, I select the two sequences, and I have here in my toolbox, I do have the little tab cloning, and I select restriction-based cloning if I want to make such a golden gate reaction. If I did not select my sequences before, I can now do it in this file here. So I can still add another one. Oh, it's too big. This one, for example. So maybe oops. Maybe you have more files to select in your cloning. So you need to select all the files that will be involved in your cloning reaction. You just say next. And here, well, open or save. I basically never save cloning files. So I would always opt for open, but this is of course up to anyone's taste. And now we get this type of window. So to make in silico cloning with a CLC, you have to look at 3 different areas. So we have a drop-down menu here, which contains all the different molecules that we have selected. So if it's a complicated assembly with many different components, you will of course have all of these here. And then you have here a little selection that says vector. For the moment, none. Change to current. So this is where we have to select which molecule shall be used as a backbone in the product. And this is always the first thing that you should do, because everything you do before will be lost the moment you do this. Yeah. So if I now start, for example, digesting this. So to digest this PCR product with BSA1, I just click on the BSA1 side here. So now I've cut it once, and I want to cut it twice. I press Control on my keyboard and click on the second BSA1 side. And now we'll have 3 different fragments. So I will have this 9 base pair 5' fragment. I will have this 13 base pair 3' fragment, and I will have the central fragment. Of course, if I want to continue with cloning, I want the central fragment, which actually represents my gene. I do not use the enzyme BPI1 now, so I just turn it off to not confuse with its presence. So I would, of course, want to use this molecule. But now, if I say that I actually have to define my plasmid at some point of time, so this is the one that I downloaded from AdGene, which I want to use to clone this PCR product. The moment I say change to current, what I did before, this in silico digestion, is gone. So always first start by changing to defining the plasmid, the molecule that shall serve as a backbone. So simple golden gate. So I digest the plasmid. Once again, I click with the left mouse button on the first restriction side. I press Control on my keyboard and I click on the second restriction side. And now again, I have 2 different. So I cut a circular molecule twice. So what I will get are 2 fragments, and I have to select the right one. So I want to continue with the bigger one in this case. So the CLC workbench will by default always select the bigger fragment if this is what you defined as the vector, and will by default always select the smaller fragment if it is defined as an insert. So this is not always correct, and if your things don't fit together, then this might be one reason. Okay, so now I have selected the right molecule, and I can now go to my PCR product, and I also digest it. So once again, left click and pressing Control, a second left click, I have the central fragment. So I can now, it now tells me that I have a target vector selection defined. I have a fragment to insert, only one, so I can clone. It's a single fragment, and now it fits. So we have the green ticks here. But if I, for example, change the orientation, it doesn't fit anymore because we have 2 different sticky ends on the 2 extremities. So now it doesn't fit anymore. So I have to turn it around. Unfortunately, there is no, right now it was correct right away, so I could have clicked on finish. But if there are several fragments and so on, there is no automatic alignment with corresponding sticky ends, unfortunately. So you have to order them by yourself to have something that is compatible at the very end. So we can say finish, and now we'll get my cloning product. So this would be now my gene GAK under control of a plant promoter with a plant terminator and contained in a tDNA for plant transformation. So I could make, for example, transgenic Arabidopsis plants expressing GAK-A. Most likely they would be just like the wild type, but this was just as an example. So the result, I can of course save it just by dragging and dropping. And I don't want to save this cloning project. What I see in this file now, if I open it in the history, is where it actually comes from. So I've made a cloning between PICH 86099, which I cut at several different sites, and a PCR product that I also cut with BSA1 at several different sites. And this gives now this final construct of 9,127 nucleotides. And this was Golden Gate. So I told you in the very beginning, the restriction sites, if everything is properly designed, are eliminated. The product does not have any restriction site. And as you can see, we used BSA1. There is no BSA1 site in my final construct. If I had used BPI1, these internal restriction sites in my GAK-A gene coding sequence would have actually been a problem. But since this was BSA1, it doesn't really matter. So this would be more or less the exercise number 1 that I put into the PowerPoint presentation. But this one says to use a eukaryotic gene, so AT1G whatever. Let me go back to that. AT1G0F9 1190. And if I want this, I can go into the Arabidopsis genome here. So you have the chromosomes. It would be on chromosome 1 because it started with AT1G. For those that are not familiar with Arabidopsis gene nomenclature. And if I search in the annotation, I should find this and only this gene. Yeah. So now I have an intron-containing gene. So it actually does make a difference whether I clone a cDNA or a genomic DNA. And I said in this exercise that you should use the cDNA. So the only reason why I'm insisting on this is that to just see the differences between these annotations. So blue is gene. It should normally also contain or encompass 5' and 3' UTRs. So maybe this gene doesn't have UTRs, or maybe they have simply not been properly detected. But the annotation gene, the blue arrow, is everything that is transcribed into the, well, unprocessed mRNA. Then there is splicing kicking in, and the result of splicing will be the green arrow. So the green arrow also encompasses 3' and 5' untranslated regions, while the yellow arrow only encompasses the coding region, so from start codon to stop codon. It's not a good example. So here we have some UTRs, yeah, and you see there is a clear difference between green and yellow. So here we have only one transcript. So especially in for example, animal genomes, human genome, you will have many different splice variants and so on annotated on the genome. But if we want to have the cDNA, so what I can do is just use the coding sequence, the yellow-greenish, well, yellow arrow, make a right click and say that I want to open the annotation in a new view. Since the annotation is on this strand, it will open it like this now. Well, it will always open it from 5' to 3' in the sense of the orientation. And what is nice, it actually maintains this little information that there was an intron at some point of time. So if I make a right click and I say edit annotation, I will now see that some fragments, some parts were joined, and this is what we actually see here in the graphical view. So again, the exercise was to clone this as a PCR product into this plasmid. So what I have to do again is to add these adapters. So it was AATG on the 5' end, GCTT on the 3' end. You can quickly do that, and BSA1 sides. So it would be TC, and then another A to establish the sticky end. Why I'm doing this is to show you that now I have actually screwed up my annotation. So my annotation, coding sequence, and so on, they should start at the ATG and not here. So I have to correct this. Since I only want to work with this, let's say, with plasmids in the future, I don't need these original annotations about gene anymore, I would say, and mRNA, so I delete them. But at least the coding sequence annotation. So now it will not do what it's supposed to do when I show the translation, because I actually shifted the reading frame. So I want to correct this. If I select the beginning of the gene, yeah, so I actually used capital letters and small letters to make the difference here. So if I mark the beginning of the gene and put the mouse over this, I will see in the bottom right corner of my software, so down here, whoops, down here, that my selection starts at position 11. So what I can do is make a right click on this annotation, and I say this doesn't start at position 1, it starts at position 11. There I go. And now you see that my annotation, at least on the 5' end, starts properly with the ATG. I have the right reading frame and so on. And here it stops with a TAA. Makes sense. So I can now also add my cloning overhang and so on. I do this differently than last time. So on the right-hand side, it was a GCTT. So I first add this on the top strand, because this is the way I always read these overhangs. Okay, I have GCTT added. Now maybe I have my trouble inverting the restriction side and so on. So it's GGTCTCN. So what I can do is I add it on the bottom strand, yeah? So if I select this here and not the top strand, then I will add, whoops, on the bottom strand in 5' 3' direction. So this actually means that as soon as I click edit selection here, this is the 5' end, so this part on my sequence. So I now have to add TT. I said always these two useless nucleotides, let's say, or hold on to me nucleotides, GG, TC, TC, and now I put an A for fun. And there I go. So now, if I have BSA1 displayed, I have an AATG overhang that is cut here. I have a GCTT overhang that is cut here, and I did not have to invert the restriction side in my mind. Again, if I want to make primer, so now it's Arabidopsis, it's actually in many instances quite GC poor. So maybe I want to use a bit more here, 22 nucleotides up. So you may have noticed, so I selected this on the bottom strand, and this is why it was directly, when I did the right click, tac, add annotation, what was proposed to me here was the minus orientation on the DNA. Here the same, let's make a primer. So yeah, well, should work. So this would be my full primer. And this would be now my primer number 3. There we go. So once again, I put these in my Excel list because I will want to order them maybe tonight. And this is the easiest way. So double click on the arrow on the annotation. Control C for copying will get me the right sequence in the paper press. And now then paste it into Excel. So this is basically how, well, how you add annotations, how you manipulate your sequence without losing orientation, I would say. So I just copy the name from here. Let's say that I want to, I want to also save this here. So it's Arabidopsis thaliana, CYP78A8. And I can add its cDNA. And I add it's my PCR. Okay. Again, I don't have any internal BPI1 site, so I could just proceed with the cloning as I did before. But let's just for fun do something different. Let's try to make a translational fusion between this gene GAG-A and this gene that is called CYP78. So I want to make a translational fusion, and let's say I want to put a serine glycine linker of serine alanine, 3 serines, 1 alanine in between. So how can I do that? And I want to clone all of this into this PICH 86099. So I would first of all have to look into a codon table to find out what the codons now will be serine and alanine. I said 3 serines, 1 alanine, I think. Here we would have one. Serine. For serine, I have a whole lot of different codons, and alanine is also everything that starts with a GC is an alanine. So let's say I want to add 2 of the amino acids, or well, the nucleotides coding for 2 of these amino acids on GAG-A and 2 on the other side to equilibrate the primer links. So I start here. So now if I want to make a translational fusion, first thing I have to, of course, think about is that I don't need a stop codon. So this has to be eliminated, the TAG. I turn on the translation to make things a bit more visual. So this has to be eliminated. And I also don't really need this. So maybe I can just keep the restriction site here and the additional nucleotide, but all this has to now be replaced. So we have a CAGC, which is a serine at the very end of GAG-A. And I said I want a serine alanine linker. So now, since there is a serine at the very end, this is maybe a bit weird, but let's still put the full sequence. So I would need 2 codons coding for serine following this AGC codon. Yeah, so I use this, what was the old overhang, and I say I select this and I say with a right click, edit. And I will need 2 serines. Let's use TCT and TCC. TCT, TCT, TCC. And we could, for example, use this TTCC. It's not good, but it's actually should be okay. As a new overhang. So we would have now here AGC, the last serine of GAG-A, and then 2 new serines that would represent my linker. And now I have a new cloning overhang, TTCC. What does that mean? So this can be used to basically glue the 2 PCR products together in the middle in a new cloning. So now this would not be this primer anymore. It would now have to be called 005, I think. And I have to save this as a new product. And now I said I want to have this followed by the CIP79 from Arabidopsis. So this overhang will still be fine for cloning into the plasmid, but we have to adapt this one. So this was basically something that we have freely chosen. So if I just change the codon that codes for serine, I go back to the codon table. I could also use TCG. Then this will of course give a different overhang. Yeah, we have to repeat this on this side here. So it has to be in frame. No, it doesn't have to be. The result has to be in frame. So once again, we can keep the TT, GG, TC, TC. And now also the A. But this one here, this one doesn't belong to our into the new scheme. So I just click the A and I first put the new cloning overhang. So I just put this. The things will fit together, but they will not be in the right frame. Actually, it will be in the right frame, sorry. But still, we still want to add another serine and we want to add an alanine. So I go back to the codon table and we use now an AGT codon, why not? AGT, and for alanine, a GCT. Okay, so our BSA1 here will cut TTCG. Here it also cuts TTCG, so these extremities fit together. And this one fits to the plasmid, so we could normally clone this. So let's just see what the result gives. So I just wanted to show what the cloning with two different fragments looks like. Up. So see, I already have something selected here. Maybe I've forgotten that I want to make this translational fusion. Just wanted to show you that even once you're in this cloning view, you can still make a right click somewhere here. And you can say that you want to add a sequence. So I can still add this third sequence, which I forgot in the beginning. I don't have to start all over again. So I insist again. So if you want to do a cloning with CLC, first thing to do is to define the plasmid that you want to use as a backbone. So it's again this PICH. I say change to current. Everything I do before doing this will be lost. I digest this with BSA1 by pressing Control and clicking on the restriction sites. I have my first PCR product. I digest it again as well. I have a second PCR product, which I also digest. And now normally this should have worked. I have saved this as a different time point before changing this little last letter. Now it will work. Sorry for that. So remember that I changed the serine codon at some point. So now it works. So I just would have where I just didn't save the very last changes. My mistake. Ah, yeah, because I did these changes in the file copy that was still actually this one here. And I had made a new copy in between. Yeah, stupid mistake and actually weird handling of the software, of the files by the software sometimes. Anyways, now it works. And now I can actually change the order here. So you see, okay, there is no button to say, try to make it fit. I have to actually find out how it works. And for that, I need some meaningful names because otherwise it's just trial and error if I don't remember what I did. So now I just save this again up. close everything. And what I should get now, so I can check whether the stuff I did is correct. So I wanted to have a translation of fusion, so everything should be in the right reading frame, in the same reading frame. So what I can do, because I did not properly annotate things here, yeah. So I can now either I make a new annotation which goes from the beginning or from the end of the Arabidopsis gene to the beginning of the bacterial GAG-A gene, or I can show all reading frames. So I can make a right click on all this. I can say add annotation, GAG-A 78 fusion. This would be now a coding sequence, so CDS up. And now if I select that and under nucleotide info that I actually want to see the translation, I can, so here is where they come together. So in the moment I put the mouse on the GAG-A annotation, I will only see this translated, and then the next one here. In the moment I put it on the long translation of the fusion protein, then I will see now that everything is in the right frame. Yeah. So if I go here, I see that my GAG-A stops with a serine, and then the CYP78 continues with the methionine, and which we could have actually eliminated because it doesn't make sense to keep it here. And if I mouse over the long fusion protein that I've just annotated, I see that everything is all right. So I have my serine adenine linker, and everything still is in the right frame. And I can of course also now start translating this to see what I get as a protein. That's beautiful and useless. Anyways, so that much for some more or less simple Golden Gate cloning. I could just go back to this PowerPoint. So what I did here as another exercise is that you should clone an Arabidopsis gene into a different plasmid. So PAGM1287 contains a sequence coding for GFP, I think, something like that. And then you should create a new overhang so that we have actually a fusion. And then I also propose that you do actually you were supposed to get another an M-cherry coding sequence as a brick from Edgene, and to then make an assembly of all of these into a fusion protein. Okay. And then as the fourth exercise, maybe we can just do this one. Was the elimination of a restriction site by using PCR and in a single step by dividing your gene into several PCR products. So just get this gene 81G01190. Is it still the same? Yes, it's this gene 81G01190, and I said I want to eliminate this one by PCR. So what do I do? If I want to do that in a single step, is that I simply divide the one gene into two fragments that I will then fuse by Golden Gate. And in the overhang, I will basically embed a silent mutation to eliminate the BSMB1 site. So what do we have to do first? We have to, of course, display the translation of the gene, because otherwise we will do mistakes. So I show this. So actually here where we have this BSMB1 site, we have an arginine residue. And once again, the first thing to do would be to look into the codon table and see what we can do with that. So arginine, arg here, we have quite a number of codons once again. We can, so here we have an AGA, which is used right now. We can put CGA, for example. This will, So now the BSMB1 side is gone. What we then have to do is to select 4 nucleotides that we can use as a new overhang. So I just randomly take these 4, add a notation, and I call this overhang. This is more a note to myself than anything else for the time being. So is this overhang okay? We would have in a simple cloning, so let's just say I want to put this gene ATCYP78 into the plasmid that I used before, so with a AATG GCTT overhang, but I want to also eliminate this internal site. So I would need a new overhang that is compatible with those that I have for the moment. And to check that, we can either try to compare them and to see how many mismatches there are and so on and so on. But we can also use a little online tool that New England Biolabs is providing to us. So I don't know if I put a link into the PowerPoint. I just go there quickly. So here you add your overhangs, and then the software will analyze to what extent this will result in a precise ligation or not. So we have 3 different overhangs in this one. So just AATG, didn't write AATG, simple. Then there was the 3' overhang GCTT, and now the 3rd one that I randomly selected more or less, it was CGAC. And this should normally give you such a matrix. And if it's all blue, like I have here more or less, then everything is good. And it tells me that I have an estimated ligation fidelity of 100%. So it's nice. So this would be perfectly fine as a design or as a new overhang in such a cloning. If you have more overhangs, this can become actually quite a bit more tricky. So how do I make 2 PCR products now? Both will have to contain the overhang, and everything has to start and stop with BSA1 sites. So what I can do is I take this section here, I say open selection in a new view, up, and then I also take the downstream part, and I say open selection in a new view. And what I just have to do is to add some adapters and design primers. So once again, so CGAC, whatever, I now do need a BSA1 side downstream of that. So it is N, I put A instead of that, and then GGTCTC in the other direction. So now it's GAGACC, and I put 2 additional A's. So now I have BSA1 cutting this, and my primer has to introduce these adapters. The thing is that I do have now also this adapter here, so I have a mismatch here if I use, I have a mismatch here theoretically. This is a new, this base is not present in the template when I start doing PCR. So now for having something that nicely hybridizes, I would make my calculation rather with this part. Well, I would make it with this part, not rather. And since this is very, well, many ATs, we could maybe make it done until here. I don't know. Depends also how good the PCR works. But anyways, let's just take this. This now would be my primer number 7, I think. Wrong direction. This would be the 5' part. And now here on the second one, I can make my life easy as well. I just take this this adapter sequence So that's basically my BSA1 site until including the N. So there we go. It's CGAC, and it also was sometimes the restriction sites are not shown anymore, and what you need to do is just close the file, reopen the file. Here it's also CGAC, so this fits. This is correct. Again, I need a primer, so here we have a lot of GC, maybe until here. So this would be my primer number 8. Just to show you that I didn't make any mistakes, I will quickly perform the cloning. That's the nice thing. Clonings are very fast on the computer. No need to transform bacteria. No need to wait for ligase to work. So 5 prime, tuck, tuck. Also digestion is very fast. I forgot to define the plasmid. So this is what happens. You have to start all over again. So the 3 prime has to be on the right hand side. Now everything fits, and I would have again actually the same thing basically as I had cloned here, just with the difference that here I now eliminated the internal BSMB1 site. Of course, this may not appear very logical to do so, but this is because of the hierarchical DNA assembly, which quite often requires that you eliminate internal sites. So with the system that Silva and I use, it would be BSA1 and BPI1 that are strictly forbidden, and BSMB1 becomes a problem when you want to go into very complex assemblies through different levels. Then it should also be eliminated. Yeah. Okay, so 3 last words on cloning. So this was all Golden Gate. You can of course also do different things. So let's try to find book 57. I don't know if I can download this from NCBI as a standard cloning plasmid. Only with inserts. So I'm sure it does exist somewhere, but I'd have to look differently. So I just take my local copy. So here you have actually a search field. So if you don't know in which folder of your library you find the thing you're looking for, you can search for it. And I just use this one. I just wanted to show, so maybe I want to just store a piece of DNA somewhere. So what I can do is also to make a blunt end cloning. So I can open up book 57, for example, with EQR5 or with the SMAR1, tuck, tuck, and just place my PCR product in there. So let's just do that. I have book 57. I just use this 5 prime construct, 5 prime fragment. I say finish. Book 57 is my plasmid, and I said it's either, for example, SMAR1 or EQR5. That will do the job here. EQR5, SMAR1. So I did not check this before, so I should not have any internal sites for any of these in my insert, which is the case. So I would in this therefore prefer SMAR1. So I have already defined book 57 as my plasmid. So I would just open it once with EQR5. This is inside a LUGZ marker gene. So again, we have blue-white selection for successful cloning. And then I have the second one. The only point I wanted to make here is that now I have to add as a fragment. Yeah, so you don't have this thing here activated if you only have one molecule selected, we cannot digest it. So what you do is you say add as a fragment, because it just stays as it is when it has been amplified by a hi-fi polymerase. Now we can clone. So this is non-directional. It can go in one direction or the other one. It will always fit because it's blunt end, and both of the orientations would be fine in this case. So just to say that in this case, you have to use this add as a fragment command. And also sometimes you may want to use stitch for something. That's not very often. Okay. So I think with that, I've shown you this ligase fidelity viewer. And you have a couple of exercises here. You should now be able to conduct all of these. And with that, I end this second video. Thanks.